right, so up next, you may have noticed, right behind me, we have a very interesting device that somehow made it through customs to Europe. And to show you 3D bioprinting from CellLink, please welcome Eric Gottenholm.
First of all, thank you so much for having me here in Vienna. What a fantastic place to be in. And behind being with all these brilliant minds and all these amazing technology developers and people who are working with all these cool things. So I feel really honored to be here. I'm going to tell a bit about the story of how I was inspired a few years ago and what brought me to a new set of technologies that has enabled me and my team to essentially build something that continues to inspire others, but also builds the future of medicine and the ability to work with tissues and building essentially life. The first challenge that we're looking at today is there's a massive shortage of organs and tissues in the world. And in fact, every single hour, one patient dies from the lack of organs and the lack of ability to get an organ transplant. This is, of course, due to the lack of tissues out there. The second challenge we have, which we're working with heavily, is it takes a very, very long time for a medicine or a pharmaceutical drug to get developed. And it costs a lot of money. In fact, it costs more than $2 billion. And the major challenge in this industry is that the timeline for this drug is, of course, while it's very long, we realize that these new medication or these new pharmaceutical compounds, they don't work on us, but we find that out in the late stages, in the very late stages of the development. So when you go through, develop a drug, you start with testing these drugs on human cells, and then you go into animal studies, you test it on rats and primates and dogs and a few other models. And then you go into humans. And while you're testing these drugs on humans, the fact is that 8 out of 9 drugs, they fail in human trials. We need better models and a better way to develop our drugs, and we need a better way to improve the organ shortage challenge we have ahead of us. And today, actually, what we have here is a technology called 3D bioprinting. This technology enables researchers and scientists to print human tissues and organs using a 3D printer, using the combination of human cells and a novel biomaterial. And I'm actually going to walk you through a demo of one of our printers that is standing in more than 50 countries worldwide, where researchers are using this technology today. So let's see if we can get it to print. And I'm going to set up the print process, and we're going to print a small human ear. All right. So the files we have is on a USB stick. Let's see here. And to explain kind of how the process works, first of all, what you have to start with is the ink. And that's the most important component behind this material and behind this technology. So you have the ink, which is novel biomaterial. And what you do is that you put this ink, you put it in the printer, you load it with human cells, and the next thing you do is that you insert a file. And the different files that you can put in, for instance, an image or a file of a human liver or a human cornea, and then you start the printing process. And the way the printing process works is like this. So you print for instance, in this example, we print a human artery. And in that artery we have human heart cells. And if everything works out well, then these cells should live and they should thrive in this environment, and they should be able to produce the hormones, the proteins, or the collagen that they're supposed to, and essentially feel at home. And the way we look inside of the structure is actually that these human cells are beating. And that's of course showing that this technology has successfully worked for these cells, and that the environment is doing good for the cells, and the cells can do what they're supposed to. So now imagine placing a drug on these cells or on this structure and understanding what happens. Understand if these heart cells are dying, or if they keep living, or if the heart cells beat faster or slower. Now that would enable us to essentially understand at an earlier stage whether this drug is going to work in patients or not, even before going into animals. Another amazing example of the tissue that we're working with, since you can print healthy tissue, why don't you print diseased tissue? And the fact that we can actually print cancer tumors, that's something that pharmaceutical companies and researchers are using already today to develop better and more efficient drugs. So for instance, taking human cancer cells directly from patients, reproducing them, placing them inside of an ink that's specifically designed for cancer tumor environments, and then printing it. And then you can print it thousands of times. It's very reproducible, which is why this technology is so fascinating. And then of course, test the drugs on it. Another very exciting example of a technology that some of our collaborators have developed at Rice University, and a technology that we're now commercializing in all over the world, is the ability to print human alveolus. These are tissues inside the lung, which essentially enables the blood to get oxygenated. So when you breathe in, you get oxygen into your lungs, and that oxygen is dispersed into your blood, and it oxygenates your blood and then pumps out through your body. And the way that these alveoli, if you see the look through the right on the picture, they have a lot of blood vessels around them. Blood vessels that essentially distribute the oxygen, and that brings the blood back, and unoxygenated blood is then repopulated with oxygen. And so this is really fascinating. The next slide is actually on something that the researchers have been working on the last couple of years, and this was on the front page of the Science Journal, and it came out last week. Now this is how the alveoli actually works. And this is a printed environment. And you can imagine how pumping blood through that, unoxygenated blood, pump oxygen through that transparent cavity, and then being able to oxygenate that blood and test different compounds on this. Get a better understanding of how for instance, asthma is working, or other diseases inside of the lung. Or maybe even print a cancer tumor within this to understand lung cancer. But what's most important in this field, of course, and what has brought our company to existence, and what has brought us to the market is of course the ink. Just like with any other paper printer or in a 3D printer, you need a material that can sustain the environment. You need a material that you can work with. And for our purposes, this ink is where the innovation is. And we realized when we started back in 2015, so we're quite a young company, we realized that to be able to build an industry out of this, and to be able to build a company that can actually provide technologies that changes human lives, we need to have a material that's reproducible, but a material that actually serves the purpose for living human cells. So it's not magic, it's real, and what we're trying to do is that we're trying to create the microenvironment. So the microenvironment is essentially, we take this material, we look inside the body, we look inside the liver, for instance, and we look at how these cells, how this structure is built up. And there's a lot of collagen, for instance, which is essentially what our tissue is. There are a lot of blood vessels, there are a lot of proteins, a lot of other things. And what we have to do is that we have to replicate that environment as good as possible. And then we come up with the ink. But of course, as we are a startup, One of the challenges for us to get out and start offering our products to customers was that the printers that we would sell our inks for, they were so insanely expensive that essentially no scientist could afford them. Only research institutes like Harvard or MIT or the very highly prestigious institutions could afford these printers. And matter of fact, this is not a bioprinter. This is one of the first IBM computers. But this is kind of symbolizing how big and how complex these printers used to be. You know, there used to be hoses everywhere and cables, and it used to take people 6 to 12 months to be able to get started to print different tissues. And we as a company, we had a vision, and we still have that vision. And our vision is to enable these researchers to work with this really cool technology, because we understand that we have one vital component of it, which is the ink. But the challenge for us was to get this ink out to everybody and get people to start working. So we had to democratize the technology. We wanted to bring this out to the hands of scientists and brilliant people all around the world. But the only way to do that is to drop the prices so significantly that everybody can afford it. So we launched the most cost-effective bioprinter in the world. And we sold it for $5,000. And that really impacted the field, because that gave us an opportunity to get scientists from everywhere to start working with this. And essentially, in just a matter of 12 to 24 months, we already had our technology in more than 40 countries worldwide. Today, researchers in more than 50 countries worldwide are using this technology. And it's fascinating because as a company, we see that we can impact the field directly. But what's most fascinating is, of course, what these researchers and scientists are doing with it. And of course, while we're building the community out there and while we're being supportive for our customers, I'm very proud to have my team behind me. And my team essentially is composed of scientists themselves, scientists and engineers that are working with this tirelessly day and night. And we have our researchers and scientists in essentially we're located in Boston and Gothenburg, Sweden. Those are our main locations. And that's where we're working heavily with the research. We build everything in-house. We manufacture. As I mentioned, we do the in-house research, and we also do sales and marketing and everything. And that has been the secret to our success and the secret to our customer success, always being available for them. If you look at essentially a little bit of data ever since we started and commercialized our technology, before we started back in 2015, when the technology was very expensive and not very user-friendly, you can see how slowly that technology adoption rate is moving, and how slowly researchers are moving with this in this field. And it taught us essentially to be a lot more available, and it taught us by bringing this technology down to a disruptive price point and high availability, we can really scale it up. So these are some of the examples and some of the great things that our customers are doing. And here at Newcastle University, they're printing, for instance, human corneas. You wouldn't think, but human cornea is a massive medical challenge today. There are about 6 million people waiting for cornea transplants. This massive need can only be solved by rethinking the way that we're doing research and science. Another area which is very exciting is for instance, printing musculoskeletal muscles or blood vessels. And that's something that they're doing at University of Texas, Arlington. And we're very excited to be able to promote this work, what these researchers and scientists do, because for them, that's really transformational. And this really enables them to continue their research and be empowered. For instance, at University of Plymouth, they're printing human skin. And that's another exciting area which we see has seen tremendous growth. And in a matter of fact, being here in Europe, the European Union banned the use of animals in the development of cosmetic products. This was back in 2013. And while that's created a major gap in the market, it enables us now to fill that gap with this technology. Or in Perth, Australia, they're printing human heart tissue. So the areas are almost endless. And what our job is as innovators and entrepreneurs and technology scientists, we need to enable this research everywhere. And we need to make sure that people can work with this everywhere at the lowest cost. Because we don't know where the next innovation or where the next drug for cancer or the next medication for any of the diseases out there is going to come from. But what we do know is that we need to make it available for everybody. And that's what the whole journey of democratization comes from. So with that, I would like to, of course, thank you for your attention and welcome you to the biological revolution. Thank you.
All right, fascinating stuff, Eric. Thank you very much.
Thank you.
Really cool. Now, we have a few questions for you from the audience. So Michael, wherever you are, Michael wants to know, what are the limits of what you do in CellLink?
So the limits, of course, and I think that, you know, we want to print organs for transplantations.
Yes.
I mean, that's the future. That's where we see this going. And I think that one of the limitations, of course, speed of how things move. You know, you work with a university or work with a research institute, it's slow.
Yeah.
I mean, things are slow. You got to find good students. You got to get cells to grow. You got to get, you got to get into this field quite heavily, and that takes time. But I mean, one positive thing is that it's moving in the right direction. And I think as a company, what we have to do is that we have to work really closely with customers and making sure that things are working out.
All right, and we have another one from Anonymous. How far are we from printing brain tissue or an entire human brain? I think it was on the graphics. Did you see before?
Yeah.
You're actually scan printing
the
a brain. Yeah,
nice brain.
Yeah.
So I think we're far away, of course, but from printing brain tissue, we're not that far away. I mean, printing neurons, we have different inks or different materials that can be used for printing neurons and connecting them and building neural networks. From printing an entire human brain, very far.
Yes.
Very far. I don't even know what to say with number. Number of months.
And
or years.
Another question, how large can you print? So we saw your machine.
Yeah.
It looks big enough to print something like about this big, like a heart.
It can print, so you know, the print structure, what most of the scientists and researchers print today, you know, they print structures like this. So very small. But I mean, of course, you could print something that's about 10 by 15 centimeters.
Okay.
So quite large. The challenge with printing large structures today is keeping them alive. So you need to have vasculatures, and you need to have perfusion of nutrients and oxygen into it.
Because the cells on the inside that aren't bathing in the nutrient liquid start to go.
Exactly.
Yeah.
Start dying. So you need the blood vessels.
Okay. And not to put you on the spot, but how many years do you think we're away from printing kidneys?
Kidneys and organs?
Yeah.
I think we're looking in probably for the next 10, 15 years, we're going to start seeing these types of technologies being implanted into patients. And I think what we're going to see is first more easier tissues like skin or cartilage, where you see essentially kind of single cell tissues coming in, and then after that we'll start attacking the more complex challenges.
Okay.
Could you print me a body so I can feel all the excitement in the room?
He wants to know, can you print him a body so he can feel the excitement in the room?
Yeah, that's a great question. You can borrow mine for now, and then you'll feel this amazing energy. But of course, the future is we want to really help with the shortage.
Yeah.
That's what we want to do.
Okay, fantastic. Great stuff.
Great.
Thank you so much.
Thank you so much.
All right.
Thank you, everyone.
Another round of applause for Eric.